Good morning friends, good morning students. In today's class we will discuss bilirubin metabolism and heme catabolism, right? First of all, RBC after a span of 120 days, they will undergo hemolysis or breakdown to form our hemoglobin, right? And this hemoglobin will further break down to form heme and globin, right? And this reaction take place in reticuloendothelial cells of bone marrow and liver and spleen, right? And then this heme, now as this hemoglobin is broken down to form heme and globin, this globin can be reutilized for again RBC synthesis, and moreover it can be broken down to individual amino acids, right? And this heme will further break down, and the first reaction is heme is converted to via enzyme heme oxygenase to form bilirubin, right? And this reaction requires NADPH, and the ferrous iron is converted to ferric form, and there is endogenous production of carbon monoxide, right? And this biliverdin is a green colored compound, right? And it is very toxic, and it is excreted in birds and amphibians, right? But in mammals it is further degraded to form bilirubin, right? And this reaction is characterized by enzyme biliverdin reductase, right? And this reaction requires NADPH, right? And it is converted to NADP. And during this reaction, the bilirubin formed here is unconjugated bilirubin, right? And this bilirubin is not water soluble, right? This bilirubin is formed during the second step. It is transported to liver, right? And it is transported to liver via bound to albumin, and this complex is known as bilirubin albumin complex, right? And this bilirubin albumin complex, the albumin is bound via two sites. One is high affinity and second is low affinity, right? Since this 25 milligram is bound by high affinity, and this low affinity can get easily attached in and can be absorbed by tissues in various pathological conditions, right? Now this bilirubin metabolism has three steps. First is uptake of this bilirubin by hepatocytes, and the second step is conjugation of this bilirubin, and the third step there will be excretion of bilirubin to bile, right? Now coming on to this step, the uptake of bilirubin by hepatocytes. So this is the liver, and this is the sinusoidal surface of the hepatocytes, right? And the bilirubin complex will move inside via facilitated diffusion, the albumin will be stuck outside and bilirubin will permeate inside, right? And this bilirubin inside the hepatocyte is bound, and this bilirubin here in the hepatocytes is bound to two proteins, that is ligandin, and the second is protein Y, right? And these two proteins, when bilirubin is bound to them, this keeps the bilirubin in solubilized form, though it is not conjugated now. And moreover, this bilirubin, these two proteins bound with bilirubin, they will prevent the efflux of bilirubin back from the hepatocytes. So bilirubin will stay inside hepatocytes. Now, the second step is the conjugation of bilirubin. Now we will proceed with conjugation. We have this unconjugated bilirubin which is present in enter the hepatocytes. It is converted to bilirubin diglucuronide, and for this we require two molecules of UDP glucuronate, and the enzyme is UDP glucuronide transferase. So via this enzyme, unconjugated bilirubin is converted to bilirubin diglucuronide because we added two diglucuronides, and this bilirubin diglucuronide is water soluble, right? And this is known as conjugated bilirubin, right? Now this conjugated bilirubin, some part of it can enter enterohepatic circulation, right, via multiple resistance protein 2, right? And this conjugated bilirubin is again uptaken by hepatocytes via, we need two, that is organic anion transporter 1B1 and organic anion transporter 1B3. So these two proteins will help in uptake of conjugated bilirubin into the hepatocytes, right? And defect in these genes will cause rotor syndrome. Now this conjugated bilirubin will enter bile canaliculi via active transport, and for this we need multiple resistant like protein 2, and defect in this or mutation this will cause Dubin Johnson syndrome, right? And this bile will now enter the GIT tract into the ileum and colon, right? Here it is acted upon by bacterial enzymes that are glucuronidases, right? They will act on this bile to form urobilinogen. Now this urobilinogen, 90% of it is oxidized and it is converted to stercobilinogen and it is created into feces. The brown color of the feces is due to this stercobilinogen, and 10% will enter enterohepatic circulation. From kidneys, it is enter the to form urobilin, and it is further excreted into urine. The yellow color of urine is due to this uro. disorders, the absence of this enzyme. If there is complete absence, it will cause curricular Nazar syndrome type 1, right? And if there is partial absence, it will cause curricular Nazar syndrome type 2, right? And in this second disorder, that is Dubin-Johnson syndrome, there is absence or mutation of multiple resistance protein 2. And the third disorder, that is Rotor syndrome, it is due to defect or mutation in the organic anion transporter 1 B1 and anorganic anion transporter 1 B3. So that finishes with disorders. One is bilirubin glucuronide transferase deficiency. Complete absence will cause Crigler-Nazar syndrome 1 and partial will cause Crigler-Nazar type 2. In Dubin-Johnson syndrome, there will be deficiency or mutation of multiple resistance protein 2. And in Rotor syndrome, there is complete mutation of organic anion transporter 1 B1 and organic anion transporter protein 1 B3. So that finishes with our bilirubin metabolism. Thank you.